All right. My name is Mitchell Reed, and I'm going to be talking about type 2 diabetes and how the microbiome affects it, and how also diabetes treatment affects the microbiome. So, the microbiome, first off, is it's everything that's living in us that's not us. So, usually we think of bacteria, but also viruses and fungi are a part of the microbiome. But for this presentation, I'm just going to be specifically talking about bacteria. So, what I'll be talking about during this presentation is how it was discovered, the importance of getting exposed to bacteria early on in your life, the functions of the microbiome, so how it actually affects our body, the implications in diabetes, which I'll spend a lot of time on, and then I'll wrap up by talking about personalized medicine and what we can expect in the future. So the microbiome was discovered in 1880 by this man named Theodore Escherich, and the way he did it was he was an Australian pediatrician. So he was working with kids, and what he would do was he took stool samples from kids who had diarrhea and healthy kids, and he found out that they both had E. coli in their stool sample. So that was sort of the start of the idea that bacteria were actually living within us. And then in the early 1900s, uh, other scientists actually isolated bacteria that were in other parts of the body, like our mouth, nasal cavities, skin, and we sort of got this idea that bacteria were living with us. But it wasn't until 2007, with the incorporation of the Human Microbiome Project, where we were sequencing the entire microbiome at a time. Because sequencing is now, it's relatively cheaper. It's not available for everybody with easy use, but sequencing is more affordable. And I feel like as time goes on, sequencing will be a normal part of routine checkups for doctors. So what I want to note is that even though the Human Microbiome Project and all of these discoveries are recent, we've evolved to live with bacteria for millions of years. So bacteria are essential for our survival, and we should always keep that in mind. They are a part of us. And it's speculated that some sources say bacteria outnumber us 10 to 1. Others say it's more like a 1-to-1 relationship. But what many sources agree on is that we have more bacteria in our body than our own cells. So we are a giant blob of bacteria. All right, so now I'm going to talk about the importance of early exposure. And what why this is important is that when babies are in the womb, it's speculated that they're getting bacteria from the mother, but the way they're born and also when depending on whether or not they breastfeed is extremely important to how they get exposed to bacteria. And the earlier they get exposed and how much bacteria they get exposed to is greatly beneficial to their health, not only for survivability, but it's also been shown that their brains develop faster if they're exposed to more bacteria. And a lot of studies have been done on C-sections versus vaginal births. And when the baby leaves the birth canal, they're getting exposed to a lot of bacteria. So that's good. Also, mother's breast milk is tailored to their child. So really make sure that the baby is breastfeeding from the mother if the mother can. And if the mother can't give breast milk, then breastfeeding from or drinking the breast milk from another woman who can produce it would still be beneficial. So early exposure is incredibly important. And this picture in the bottom left it just shows a variety of factors that could affect the microbiome. But early exposure is really important. So now I'm going to talk about how the microbiome actually affects us. And I really like this picture because it shows that bacteria are everywhere on our body and in our body, and they're having an effect. But I'm going to specifically talk about just a few functions. So the first one is immunity. Now this is probably the most applicable to pharmacy, and it's the one that most people know about because the microbiome actually talks to our immune system and works cooperatively with us to protect us. Now the most important aspect of this that we actually apply in pharmacy is the fact that bacteria take up space. And when they take up space, they're preventing other infections from occurring because other, fungi or bacteria can't invade that space and proliferate and cause damage. And the most obvious example I can think of this is the reason why you don't give babies honey for the first year is because honey has Clostridium botulinum in it. And if their microbiomes aren't well developed and you give them honey, the botulinum can cause botulism and kill the child. So that's an example. But also in terms of using broad spectrum antibiotics and how that can lead to superinfections, it's all based on bacteria taking up space. So they're incredibly important for immunity. They also have an effect on digestion and metabolism, and this is mainly due to how they break down foods for us. Now we have about 900 to 1,000 different species of bacteria living in us, and they all have different enzymes. So they all have different ways that they affect us. And some can break down complex carbohydrates into glucose, which I'll be talking about later, but that's the gist of it. They can affect digestion and metabolism. And they also affect metabolism by gene regulation. So this is probably the most interesting to me, but bacteria actually affect brain function. Now, there have been studies done on this, but it's not conclusive as to how they actually modulate our CNS. It's just been shown that certain types of bacteria strains have been correlated to certain CNS disorders like depression. So hopefully with future research, this will come more to tuition and we'll understand it better. But that's really a really interesting function of the microbiome. So now I'm going to talk about how it's actually affecting type 2 diabetes and how type 2 diabetes is affecting bacteria. So what I did was I looked at a lot of sources, but one of them actually was a comprehensive review of 42 different studies. And what this review did was it took the bacterial composition and abundance of different species in type 2 diabetes patients, and they concluded that certain species had a positive correlation to the disease. So if you had these species, this usually meant that the person had type 2 diabetes, but also there were certain bacteria that were beneficial for treating or not treating or curing, but helping the patient. So bifidobacteria, Bacteroides, Faecalibacteria, Akkermansia, and Roseburia, these genera or the genus were known to be beneficial for type 2 diabetes patients, while Ruminococcus, Fusobacteria, and Blauschia, if I'm saying that right, had negative impact on the disease. And I'm going to talk about these in the future. But what I really want you to take away from this is that even if I'm saying these are good or bad, it's all about how much of it you have, so the abundance of the bacteria, and the composition in which they take up your body. So if you had a healthy patient who had 1% bifidobacteria and that was healthy, if that patient had 20% bifidobacteria, that could be so high that it kills them. So it all depends. And this is very general. These are just the genera we're talking about, but each species is different and they have different enzymes. So we really can't conclude if I say bifidobacteria is beneficial, there's always going to be one species that isn't. So just keep that in mind. And lastly, Ruminococcus, Fusobacteria, and Blauschia, they have a positive correlation to type 2 diabetes. And it's been shown that bariatric treatment and surgery and the use of metformin actually increase this. So we get into a weird gray area where is it that they have a positive correlation to type 2 diabetes because they can cause type 2 diabetes, or is type 2 diabetes treatment causing the proliferation of these bacteria? So this is really inconclusive. It doesn't tell us much. But now I'm going to talk about how the microbes actually work in type 2 diabetes, how they can be beneficial and harmful. And the first major topic I want to talk about is inflammation. So we know that inflammation is a huge issue with type 2 diabetes patients. So it would be beneficial if we had bacteria that can reduce inflammation. And there are bacteria that do that. And the way they do this is they increase the essentially the expression of anti-inflammatory cytokines, and they decrease the expression of pro-inflammatory cytokines. So things like Bacteroides, Progeles, Akkermansia, Mucinophylla, and Lactobacillus plantarum are beneficial, whereas Fusobacterium and Ruminococcus do the opposite. So they actually cause more inflammation, and this is a problem. So with this knowledge, if we can sequence the microbiome and figure out how we can manipulate these bacteria to benefit the patient, that could be helpful for them. And this is future research at its core. Now, I do like this picture in the bottom left because we do know that nutrition plays a huge part in the development of disease. So, for example, in diabetes, if you eat a lot of sugar and you get just problems with insulin because of that, the sugar itself not only causes problems with the insulin sensitivity, but the sugar that you're feeding your body is also feeding your microbiome. So the bacteria that feed on that sugar are going to excel in your body. They're probably going to grow in abundance and in composition. And whatever that bacteria does in your body, it's now going to be exacerbated. So if the bacteria is harmful for your body and let's say it causes inflammation, now you're feeding that bacteria tons of sugar, so you're causing even more inflammation, which is going to make your type 2 diabetes even worse. So when we're thinking about food, we need to think about, well, the food's going to negatively affect your body, right? If you're eating a ton of junk food, but it's also going to change the composition of your microbiome, which can also impact your body. So that's really important. And I thought that was really cool. Now I want to talk about gut permeability. So diabetic patients have a lot of gut permeability, and the easiest way to explain this is in your gut, if it's more permeable, there's a higher likelihood that certain bacteria and toxins are going to be able to get into your bloodstream. And we know that's not a good thing because this can lead to things like septicemia and even death. So what certain bacteria can do is they can modulate gene expression to actually create more tight junctions to reduce that permeability. So Akkermansia mucinophylla, which we've seen before, has a wide variety of ways to do this. But just to keep it simple, modulates gene expression, increases tight junction formation, reduces gut permeability, is good for the patient. So this is another really important aspect of bacteria that can affect type 2 diabetes. Now, lastly, I want to talk about glucose metabolism. We know that glucose and carbohydrates are a huge problem with type 2 diabetic patients. And certain bacteria can reduce glucose levels for them. So, for example, if we look at Bifidobacteria lactis, what this does is it modulates the expression of the liver. And a lot of these bacteria, because they have different enzymes, they almost function like mini livers because they can metabolize certain drugs, certain foods, they can harness energy from foods. They do a lot of things, but this bacteria in particular, it modulates the essentially the metabolic phases of our body. So it decreases glucogenesis, right? So that's going to decrease glucose, but it also excuse me, it increases glycogen synthesis. So whatever glucose we have, we're going to form that into a polymer, and this is going to reduce blood sugar, which is really beneficial for the patient. Now, this is probably the most interesting thing I found about Akkermansia, but Akkermansia mucinophylla, which is a glorious bacteria, it can act like an alpha glucosidase inhibitor. So we learned about these drugs. What they do is when you ingest complex carbohydrates, you have an enzyme called alpha glucosidase, which will break that down into glucose, and then we absorb the glucose into our body, which raises our blood sugar. These bacteria prevent the breakdown of complex carbohydrates. So instead of absorbing that glucose, it's just going to be excreted fecally. So messing with the abundance and concentration of this bacteria has a wide variety of beneficial effects that we should explore. And that's all I want to talk about for the glucose metabolism. Now, to wrap up for the diabetes portion, there is this drug-microbe relationship. And this is really interesting because it's a two-way street. The microbe can affect the drug, and also the drug affects the microbe. And if you look at the top right up here, this is just for metformin. And metformin probably promotes a lot more microbiota growth than this, but this is just what's been found. And if you see metformin promotes Akkermansia muciniphila, I've just spent a lot of time talking about how important that is for treating type 2 diabetes problems. So not only is metformin going to benefit the patient, but it's also increasing the proliferation of bacteria that's also going to benefit the patient. So this is really useful information. And then on the other side, this picture was something that metformin reduces the bacteria count. There's a lot of interplay between this, but this is just really important. So the way metformin can proliferate these bacteria is really good to know. Also, the bacteria can, like I said before, they function like mini livers, so they can actually metabolize drugs. Now, if we think about a prodrug and we digest it, it gets absorbed, and then it has to go to the liver to be activated. Well, if we digest it and certain bacteria strains can metabolize it and activate it, this is going to reduce the effectiveness of the drug, because the moment it gets to the liver, it can be cleared, right? It doesn't have the first pass metabolism, essentially. So we need to keep this in mind when we're thinking about drugs. And I'm going to talk in a little bit about how this would actually look in practice. But bacteria can affect the pharmacokinetic properties, and that's what the point I want to get across is. So now I want to talk about personalized medicine and what to look for in the future. Now, I did talk about how all the benefits of bacteria and how some can be harmful, how they can affect pharmacokinetic properties. And this is a really hot spot for research. Now, I'm kind of bummed. If I was born 10 years in the future from where I was, I might actually have a class on the microbiome, because I feel like the microbiome itself is going to be a huge area of research that is the next step in how we're going to give patient care. And not only is it going to give pharmacists more to do for their jobs, which is good, but it's going to add another level of complexity and a way that we can help our patients in the future. So I'm really excited about the microbiome. But just to give sort of a hypothesis about how I think this is going to play out in the future is, if we follow this down here, we're going to try to correlate certain bacteria to certain diseases. So if we had a patient group that had a certain disease like diabetes, we can sequence their gut microbiota to figure out exactly what's in there, and then look for similarities, like, oh, all people who have diabetes don't have bifidobacteria. That's really weird. And then we could think about, like, what does bifidobacteria do? How could that be beneficial or harmful, etc. But if all people with diabetes had something like, I don't know, lactobacilli, and it's a very certain species, we would isolate that bacteria, and then we would culture it and see how it affects certain medications and therapy that diabetes patients would be on. That way we can figure out, okay, well, we know diabetes patients are going to take this medication, but how is the variability in the microbiome going to affect how they react to the medication? And then depending on their interactions with, metformin, for example, drug therapy would need to be modulated. So this is the whole personalized medicine. We're taking the idea that the microbiome, and genetics as well too, can affect the way that drugs are processed in their body, and the pharmacokinetics are going to change based on that. So we would need to modulate therapy to accommodate those patients. And then lastly, just to hammer it home, the composition and abundance of the microbiome is always taken into consideration. Having too much or too little can be a problem. So we really need to have a well-rounded abundance, and maybe in the future we can change the composition and manipulate it somehow. But that would be an area for future research. So lastly, We all know this is going on, antimicrobial stewardship. This is where scientists, they have an importance to not be stingy, but they need to be careful with how they use antibiotics. They want to steer away from using broad spectrum antibiotics unless they absolutely have to, because not only does this foster drug resistance, which is a huge problem for certain disease states like MRSA, C. diff, but also it can just totally butcher the person's microbiome or the patient's microbiome. So when we're using antibiotics, we need to think about, risk versus reward. Is the patient like dying? Do they need a drug right away? Then okay, then we might want to give them a broad spectrum. If we have time to figure it out, run lab tests, let's try to be more selective in the antibiotics we give them. Not only will this, lower the chances of developing antibiotic resistance for certain medications, but also it preserves their microbiome. And I just spent a ton of time talking about how important the microbiome is. So just to wrap up, I think this is really important. And antimicrobial stewardship should be something in the minds of every healthcare professional who is prescribing or monitoring drugs like pharmacists. So in summary, I talked about how the microbiome was discovered in 1880, but we've evolved with this bacteria for millions of years. The Human Microbiome Project really took off in 2007. So this is a relatively recent scientific discovery that I hope we further research and learn more about the importance of the microbiome as time goes on. Bacterial composition in abundance is important. Hammered this home. There's not much more to say about that, but it affects our physiology, our prevention of disease. We think about babies and botulism, superinfections. Utilizing certain species in type 2 diabetes could be a role in future research. Now, we do have a lot of drugs, drug therapy, modifications, but if we actually know about how species can metabolize drugs or how they can affect blood glucose, we might want to modulate their microbiome if we can. I'm not sure how that would look like. I don't know if we would inoculate them with certain probiotics, but that's an area for future research. That's pretty exciting. And then sequencing a patient's microbiome. I feel like this could be a common tool. Like when you go to the doctor and you get a routine checkup, they check your height, they do a physical, getting your microbiome sequenced might be routine in the future. As crazy as that sounds, I'm looking forward to it and I hope it happens. And then we do need to practice evidence-based medicine and really be selective about when we use strong antibiotics because the microbiome is incredibly important. With all that being said, thank you for your attention. And if you have any questions at all or want to discuss anything I said, email me at read3055@pacificu.edu. It's a beautiful sunny day. I hope you're staying well despite these COVID-19 things going on. But yeah, email me if you have any questions. Here are my references. And thank you. Have a good one.